Welcome back to my channel Knowledge Chronicle.
Today, we're diving deep into the fascinating world of recombinant DNA technology, a groundbreaking innovation that revolutionized genetics, biotechnology, and medicine. But this isn't just a surface-level explanation. We're going step by step, from DNA isolation to bioreactors, and even downstream processing. By the end, you'll know exactly how a small fragment of DNA turns into life-saving insulin or vaccines. So buckle up for this 14-minute biotechnology ride. Let's begin. Recombinant DNA technology is a method where genes from different sources, even different species, are combined into a single DNA molecule and inserted into a host organism. This host can then replicate and express the inserted gene, producing the desired protein or trait. Imagine taking the gene for insulin from a human and inserting it into a bacterium like E. coli. That bacterium now becomes a factory, producing insulin just like a tiny bioreactor. But how exactly does this magic happen? Let's break it down step by step. So, all living organisms store their genetic information in DNA. But you know, DNA doesn't just float around freely, it's actually hidden inside cells, and it's tightly packed with proteins. To access it, we have to break open the cells. For bacteria, we use enzymes like lysozyme. For plants, it's cellulose. And for fungi, we use chitinase. Next, we need to remove contaminants. RNA is taken out using ribonuclease. Proteins are removed with protease. And well, other molecules like polysaccharides and lipids are eliminated using specific treatments. Finally, to get pure DNA, we add chilled ethanol, which causes the DNA to precipitate as white thread-like strands. Example: this method is used in labs when isolating plant DNA for genetic modification in agriculture. To insert a gene of interest, we must cut the DNA precisely. For this, we use special enzymes called restriction endonucleases, tiny molecular scissors. Each restriction enzyme cuts DNA at a specific sequence. For example, the enzyme E. coli cuts DNA at the sequence GATTC. This process is also done on the vector DNA, a carrier that will help deliver our gene into the host. How do we know if our cutting was successful? We use agarose gel electrophoresis, where DNA fragments are separated based on size. Since DNA is negatively charged, it moves towards the positive electrode, and we can visualize the fragments using dyes under ultraviolet light. After cutting both the source DNA and the vector, we ligate or glue them together using an enzyme called DNA ligase. This step forms a recombinant DNA molecule. The gene of interest is now inserted into a plasmid vector. For example, the insulin gene can be inserted into a plasmid that contains antibiotic resistance. This plasmid can now enter a host organism, usually a bacterium. Sometimes the gene fragment is just too small or, we need more copies of it. That's where PCR comes in. PCR, or polymerase chain reaction, can actually make millions to billions of copies of a specific DNA segment. So, how it works is it uses primers, which are short DNA sequences that match the start and end of the target DNA. A thermostable DNA polymerase, taken from Thermus aquaticus, synthesizes new strands. Through cycles of heating and cooling, the DNA is denatured, annealed with primers, and then extended. For example, this technique is used widely in COVID-19 testing, where the viral genetic material is amplified to detectable levels. All right, so now that we have our recombinant DNA, it's time to transfer it into a host cell, usually a bacterium like E. coli. But, the host must be made competent, meaning it can actually accept foreign DNA. This is done through a few ways: heat shock treatment, chemical treatment with calcium chloride, or sometimes by using electroporation. That's a short electric pulse to open pores in the cell membrane. For example, if our recombinant plasmid carries a gene for ampicillin resistance, then only those bacteria that have successfully taken up the plasmid will survive on an ampicillin-containing plate. This acts as a selectable marker. Once inside the host, the recombinant DNA must be expressed. That means it needs to be transcribed and translated into a functional protein. This actually depends on a couple of things: the presence of promoters and regulatory sequences, and also the host cell's compatibility with the gene. For example, let's look at the production of human insulin in genetically engineered E. coli. Here, the insulin gene is expressed and the protein is harvested. Producing small amounts of protein in test tubes is fine for research, but not for commercial use. That's where bioreactors come in. Bioreactors are large vessels, usually anywhere from 100 to 1,000 liters or even more, where cells are cultured under optimal conditions for massive protein production. There are a couple of main types. The stirred tank bioreactor is the most common. It's equipped with stirrers, an oxygen supply, and controls for pH and temperature. Then, there's the airlift bioreactor, which uses air bubbles instead of mechanical stirring. So, for example, insulin, vaccines, and growth hormones are all produced in bioreactors using genetically engineered microbes. Once the protein is produced, it actually needs to be extracted and purified. This whole process is called downstream processing. The steps here include first, separation of cells from the culture medium, then purification of the protein using methods like chromatography, filtration, or even centrifugation. After that, formulation with stabilizers and preservatives. For pharmaceuticals, clinical trials are done to ensure safety and efficacy. And finally, quality control testing. So, for example, in insulin production, downstream processing is what makes sure that the final injectable product is pure and safe for diabetic patients. This revolutionary technique honestly has countless applications, such as medicine, for instance, insulin, vaccines, gene therapy. So in agriculture, we have things like pest-resistant crops such as Bt cotton and even drought-tolerant varieties that really help farmers deal with tough conditions. In industry, enzymes are actually used in detergents to make them more effective and, also in producing biofuels, which is pretty cool. And in research, scientists study gene functions and, they even create transgenic animals to better understand biology and develop new treatments. And that, my friends, is the complete journey of recombinant DNA, from isolation to large-scale production. This technology has transformed how we treat diseases, grow food, and understand life itself. If you found this breakdown helpful and easy to understand, then don't forget, your one like, share, and subscribe will help us create more valuable content. Until next time, keep exploring, keep learning, only on Knowledge Chronicle.